Welcome back to another episode of From Code to Cure, Ainnocence webinar series exploring how AI is transforming the future of medicine. So in today's episode, we will take a deep dive into one of the most fundamental challenges in drug discovery and a new approach that may be changing how we solve it. We will be talking about destructive drug discovery and how sequence-based AI is speeding up and expanding the search for the new therapeutics. My name is Roger Wen, and joining me today again is Dr. Pan, the founder and CEO of Ainnocence.
Thanks for having me. This is a topic I'm really passionate about. It's at the core of what we do at Ainnocence, and I think the science is genuinely exciting turning point as well.
So, just to all of our audience, this is a pre-recording session, but we would love to hear your questions and the reactions in the comments area. Whether you are a researcher, a drug developer, or just curious about how AI and biology intersects, please drop your comments below. So, Dr. Pan, let's start with the problems you are trying to solve here. So, drug discovery has always been a difficult and expensive problem challenge. How do you frame this challenge?
Yeah, the scale of chemical space is almost incomprehensible. That's the major challenging. For example, for small molecule, we're trying to find this one molecule from a vast ocean of space as big as 10 of 60 order of magnitude of number of molecules for different type of bioactivities. And then biologics the same, the antibody therapy, we need to select one out of 10 billion at least. So, which is really mission impossible if you just screen them in wet lab. So, in the conventional sense, the wet lab is doing high throughput screening with really low, like blind hit rate, about 1 out of 10,000 early hits. And also, they have to go through tens of dozens of different variety of wet lab to be able to really get the best candidates selected. So the average success rate for these conventional methods is about 8 to 12% from beginning the phase 1 to 3. So most of the project would be failed. So the true problem is multi-objective optimization, which conventionally they can only optimize one parameter at once, but also when they optimize the second and third, the first one may be compromised. So that's the true issue, and for the conventional discovery approach.
So, if that is a conventional approach, do you think AI can help for this kind of challenge?
Yeah, definitely. I think the advantage of AI or any computational methods is able to really do virtual screening on a scale which not just for a single point optimization, but at the same time multi-pharmacology profile optimization. So we'll be able to really search the vast ocean of chemical and sequence space with multiple AI models to confine that chemical space into really the drug-like window. And then that multi-parameter optimization is the only AI can do and conventional method cannot do. So that people started to really doing it. And at the same time, you probably heard AlphaFold, which is a structure-based AI model that try to predict all the structure of all the therapeutic targets and the structure of basically all life forms, proteins. And then this is a good achievement on understanding its function and structure. But at the same time, there's some limitations.
So, what are they? What are those limitations?
So first of all, the all the structural predictive AI are trained on we call it the structural biology data are from either crystallography or cryo-EM. They are actually extremely low temperature and frozen state of proteins, which is not physiologically relevant, which is in solution and 37 degrees Celsius. So in that case, the training data is kind of biased. And then also it could only predict the regions of protein structures that are highly static, which a lot of important biological regions of different proteins, especially those flexible ones, those regions interact with drugs are actually missing. So which results the model cannot really accurately predict the all the regions of the protein targets, and at the same time all the structure itself is being biased against very low temperature configurations. So that's the first limitation. The second limitation is the cost of both data generation for training data of those assays for structural biology data acquisition, and another one is also very expensive to compute based on the 3D structure to do any molecule screening. Because if we do a molecule protein-protein interactions, for example, over 3D configuration in a water molecule in a water environment, it's almost impossible to screen anything bigger than 100 candidates. So, but if you wanted to see, let's say, 10 billion candidates, that's how our B cell be able to compute and produce that many antibodies by nature. So that's almost impossible to mimic the natural antibody production process and screening process in our own body. So that's kind of always the limitation for the structural biology type of approach. And on the other hand, there's other type of training data are way less expensive. For example, structural biology data are ranging from like $100,000 per data point, but other like activity and binding assays less than $10,000. I'm sorry, $1,000 or even less than $100. And some data point are also even getting cheaper using microfluid technology, which the single cost of the each data point could be less than $1, which makes the training data in the other type of biologic space are much easier to acquire and much more abundant. So which give us rooms for other type of methods, such as our sequence-based AI.
That's interesting. I heard a lot of limitations and I heard sequence-based AI. Can you walk us through what you mean by sequence-based AI?
Yes. So in the biological space, as we know that our all life forms are all the information encoded in DNA. And DNA through transcription, it passed on that information to RNA. And RNA through translation passed on that same information to protein. So from DNA, RNA to protein, their primary structures are a 1D sequence, which is highly accurately measured, sequenced, and also they are always there no matter what conditions. They stay in the same coding structure, which is a string of a long, 1D thing. So in that case, we know that all life form come from there. And 3D structure is also generated from 1D because once we know the 1D, we know the 3D. But we not just know the 3D, we know all the other downstream biological effect from its structures, from its activities, from its stability, its half-life, and also many other bioactivities. They're all directly related to the 1D sequence. So all those different layers of dimensions, even in the time domain, the time dimensions are all encoded in the one-dimensional space. That's why actually that's the origin of the life information with all the information already hidden in the sequence. So it's just how we could decode this information. And in order to do so, we could, like I said earlier, we have different type of experiment measurement of those how those molecules behave once we put them in a biochemical or cell biology environment or even in vivo environment. They were started to fold, interact, and everything. But at the same time, all we can always sequence and measure the sequence 1D sequence with no error. It's almost 100% accurate. And at the same time, all the binding events, all the other cell biology events can also be measured cheaply and accurately. So the sequence-based AI that Ainnocence provides is directly we are taking the sequence input and then approximate and training on all those labels on the biochemical events bypass the 3D folding and all the domestic details. We only need to know the amino acid or DNA sequence, which is encoded in English letters directly, and then be able to approximate wet lab experiment data very good. And all the experiments we're training against all those labels and also the FDA submitting packages that all the molecule has to measure anyway. So which make it the entire journey of drug discovery more end to end, more translational to clinical stage requirement. So that's kind of the general overview of how we tackle this problem.